Question 61. Question 61 asks that the diagram shows an important concept in the genetic implication of DNA. Fill the blanks A to C. Now here in diagram they have given this, then they forming a mRNA, and here A is written. Now here obvious A is showing a conversion of DNA to mRNA, means A is nothing but a transcription. Now from mRNA it is forming a protein, and in protein B is given, means B is a translation, and later on it is showing a proposed by, and here they have written C, and C is nothing but Francis Crick. So according to this option A is correct answer. Question 62. Question 62 asks that self-replication is occurring. Self-replication or self-duplication or automatic replication does not occur in case of enzyme, lipid or protein. It occurs in case of DNA. So option A is correct. Next is question 63. Question 63 asks that removal of intron and joining of exon in a defined order during transcription is known as. So you know that the removal of intron and joining of exon is known as splicing, which is a part of post transcriptional modification. So option D is the correct answer. Next is question 64. Question 64 asks that Griffith have performed a series of experiments on. Now Griffith performed transformation experiment, and this transformation experiment proved DNA as genetic material transforming chemical. Now for this they have used bacteria, that is Pneumococcus pneumoniae and mice. So they used pneumococcus. So option A is correct answer. Question 65. Now question 65 asks that which enzyme or enzymes are responsible for transformation of lactose to galactose. Now lactose is broken up into the glucose plus galactose. Now which enzyme is responsible for this in E. coli culture medium? So you know that this breakdown is carried out by beta galactosidase enzyme, which carry out a breakdown of lactose. So option A is correct answer. Next is question 66. Question 66 asks that in typical nucleosome, if adenine is equal to 10, then how many guanine are present? So you know that in typical nucleosome DNA takes 1/3 ton, means one complete, and another ton which is a 3/4 of the 3/4 part. So this much touch DNA take around the histone protein, and in this 1/3 ton 200 base pair DNA is present. Now if 200 base pair means 400 bases, and out of 400 bases adenine is 10, means thymine is also 10. So remaining, so if we remove this 20, the remaining is 380 is distributed among the guanine and cytosine. So guanine are 190 and cytosine are 190. So guanine are 190 according to this. So option C is correct answer. Question 67. Question 67 asks that the lake of Huron consists of. Lake of Huron consists of. Now lake of Huron consists of option A 4 regulatory gene only. No, there is only one regulatory gene. So it is incorrect. B 1 regulatory gene and 3 structural gene. Yes, 1 regulatory and 3 structural gene. C 2 regulatory gene and 2 structural gene. It is incorrect. 1 regulatory gene and 3 structural gene. And D 3 regulatory gene and 3 structural gene. So it is also incorrect. Now here there is only one regulatory gene, that is I gene, which code for repressor, and 3 structural gene, that is LEC-Z, LEC-Y and LEC-A. So according to this, B is the correct answer. Question 68. Question 68 asks that blank is common for both DNA and RNA, but blank is present only in DNA. Now you know that according to option, guanine is a nitrogenous base. Guanine, if we simple consider the DNA structure, guanine, cytosine and adenine, common for DNA and RNA, but thymine and uracil are not common, as thymine is present in DNA and uracil are present in RNA. So according to the question, the common for both DNA and RNA is guanine, and blank is present only in DNA, so it is a thymine. So option C become correct answer. Question 69. Now question 69 asks that A assertion is the process of splicing represents the dominance of RNA world. Yes, it is correct. And R is given as the presence of intron is reminiscent of antiquity. Yes, reminiscent of antiquity means intron represent the ancient RNA world. So it is also correct. So both A and R are correct. So A is correct option, that A and R are correct. Question 70. Question 70 asks that X-ray diffraction data proposed by. Now X-ray diffraction data proposed by Morris, Wilkins and Rosalind Franklin. So option D is correct answer, but they have worked on the X-ray crystallography technique, and they have developed image. On the basis of this images, Francis and Watson and Crick gave a DNA structural model. Question 71. At what level gene expression does not occur? At what level gene expression does not occur? A. Formation of primary transcript means hnRNA formation is occurring, means transcription is occurring, and transcription occurring means gene expression is occurring. B. Processing level. In processing level also gene expression occurs. C means transcriptional processing level. Translation level, yes. D. Replication level. At replication level, gene expression does not occur, because gene expression means mRNA and then protein formation. So So D is the correct answer. Next is question 72. Question 72 asks that dystrophin gene has dystrophin gene present on X chromosome, and it is the longest gene. The size is 2.4 kilobase pair, means 2.4 million bases. So C is the correct answer. Question 73. Question 73 asks that which one of the following is wrongly matched? So if we go with the option A is given as transcription, and transcription is the writing of information from DNA to tRNA. So DNA to tRNA, that cannot be considered as transcription, so it is incorrect. B is given as translation, using information of mRNA to make protein, so it is correct, yes. C is given as operon, and operon has structural gene, operator gene, and promoter gene. Actually operon has structural gene, operator, promoter, and a regulatory gene, but in option regulatory gene is missing, so it is incorrect. So A and C are incorrect, and option D is given as both A and C, so both are incorrectly matched, so D is the correct answer. Question 74. Question 74 asks that Question 74 asks that Balbiani rings are a sites of Balbiani rings are a sites of. First you should understand what mean by Balbiani rings. Now Balbiani rings are found in salivary gland or polytenic chromosome. Now in the salivary gland or polytenic chromosome, which was discovered in the Drosophila and Chironomus, in this what happens, they have so many strands of DNA, and on this they have a bulging structure and some fibrous structure. Now if we see this bulging structure in a detailed view, then they look like this. Now these are known as Balbiani rings. Now in this what happens, the strands of DNA that is present here, that will carry out a transcription and translation, and you will form a particular RNA and protein. So Balbiani rings are sites of RNA and protein synthesis. So option B is correct answer. Question 75. Question 75 asks that which of the following is incorrect for Hershey and Chase experiment? Incorrect for Hershey and Chase experiment. Now if we talk about the Alfred Hershey and Martha and Chase, that is a unequivocal proof that DNA is genetic material. Now in this experiment they have a 3-step process, and in this step first is infection, second is blending, and third is centrifugation. Now in option also this is given as A, this is given as B, this is given as C. Now D option is PCR, which is not involved. So this is incorrect with respect to the Hershey and Chase experiment. The full form of PCR is polymerase chain reaction.